Is we are going to go through this worksheet. You got it? Did you download it? Got it? Either digital or physical copy, one or the other? Okay, so here's how we are going to do this. This is a good review overview of all of the steps of contraction and relaxation and muscle. And what we're going to do with it is we are going to go through it and you guys are going to answer the questions. And I am arbitrarily, because the back of the room is so often ignored, I'm going to start at the back, and here's how it's going to work. I'm going to start with this gentleman, this bearded gentleman, and then I'm going to go over to Travis, and then I'm going to go backwards through the row, all the way to Tiffany, and then I'm going to go to you, and then we're going to move over. So you can try to calculate which question you're going to get. And what we are going to do is we are going to give it a shot and not be a big old baby when it's our turn, right? And we are going to realize that if we try, I will help you, I will flop, I will give you, I will give you some pointers, I'll help you if you almost get it right, but we're all going to try. So this whole story, what I want you to do is sort of listen, because although the words are going to say things, I can actually sort of direct you to the right answer as well. So I'm going to read them for you, and we're going to start with bearded gentleman, and we're going to start with number 1. So the functional unit of skeletal muscle fiber contraction. The thing that contracts that we drew on the board last time. What is the thing that contracts from Z line to Z line? What's that called? Yes, it is. This is a good start. I'm feeling good about this. Travis, you're next. Okay. The theory that explains skeletal muscle contraction is called the blank theory. Yes. Oh my gosh, you're my favorite class. I don't even know why I teach those other people. Now what we're going to do is we're going to start to try to get the muscle cell membrane excited. But before we do that, don't we have to start with the neuron? Yes? We have to start with the neuron. So the first part of this story is going to be related to the neuron, and then we will move to the muscles. So excitation of the muscle cell membrane. So we are going to have an action potential that arrives at the end of a somatic motor neuron. You guys with me? This is just synapses. It's the last set of notes. It's just how we connect it to this set of notes. So we are going to get the action potential arriving at the end of the axon of the somatic motor neuron. You are next. Now I want to know what blank channels open on the synaptic terminals of the presynaptic cell. What kind of channels are unique to the synaptic terminals?
Is the voltage-gated
It is the voltage-gated calcium channels. Voltage-gated calcium channels open on the synaptic terminals of the presynaptic cell. Now we are at C. C is the neurotransmitter, the only neurotransmitter that is relevant in this story. That neurotransmitter is released from the synaptic terminals. What is the neurotransmitter? It is. And it's released into the blank junction, which is the type of synapse between a neuron and a muscle, and it's called neuromuscular. Very good. Neuromuscular junction of the presynaptic cell. Good. D. I just released it, so it should be pretty easy. What is it? Acetylcholine. It binds to, what does acetylcholine bind to? Let's see, we are all the way over to you. Acetylcholine binds to what? We are at D. Acetylcholine binds to blank. Just throwing out there, probably. Yeah, acetylcholine receptors. Acetylcholine binds to receptors on the, okay, it's WUTA, right? Mm-hmm. Okay. WUTA, acetylcholine binds to receptors on the blank portion of the sarcolemma. It's that super curvy portion that has all the acetylcholine receptors on it. Do you remember what it's called? No, inside, that's the inside part. It's a super curvy part. Do you remember about? Do you remember? Motor end plate. I'll come back to you, please. Motor end plate portion of the muscle cell membrane. Okay, does that make sense? Which is also called the sarcolemma. The muscle cell membrane is also called the sarcolemma. Okay, these protein receptors that we just found to are also acetylcholine-gated channels. Yes, sodium channels, exactly. Acetylcholine-gated sodium channels. Opening these channels, WUTA, I'm going to go back to you. Opening these sodium channels causes a blank polarizing graded potential and excitation of the sarcolemma. So we are at F. Yes, depolarization. Good. Quite a bit easier than motor end plate, right? So a depolarizing graded potential and excitation of the sarcolemma. Now we are at G. If threshold is reached, let's see where we go after that. I already got you, so you. If threshold is reached, an action potential begins on the sarcolemma and travels deep in to the muscle fiber via little blank, big blank. T tubules, exactly. Via T tubules. Then the action potential arrives at the thing that's right next to the T tubules where you store calcium. Do you remember what that was? Right next to the T tubules. So you're going down the T tubules, and it's right next to it, it stores calcium. You know? Okay, we'll come back to it. Do you know what's called? It's a sarco something. Yes, sarcoplasmic reticulum, the SR, right? Had to give you the one with the most syllables of all. The sarcoplasmic reticulum, or the SR. So if threshold is reached, the action potential begins on the sarcolemma and travels deep into the muscle fiber via T tubules. Then the action potential arrives at the sarcoplasmic reticulum. And only after that can you get to the contraction phase, because we are going to go back. Action potential causes what to be released from the SR? Action potential causes, Ethan, what to be released from the SR? Calcium to be released from the SR. Good. Calcium binds to the sarcomere at a small protein fiber called acetylcholine. What? Calcium binds to the switch. Do you remember which protein is the switch? No, it's a protein, and it starts with tro. Do you know what it is? Troponin, exactly. It binds to troponin, which is the switch. Small protein fiber called troponin, which is the on-off switch. With calcium bound to troponin, that other tro protein, going to you, that other tro protein, which is the barricade protein, will be, which prevents cross-bridge formation, is lowered. What's that tro protein? It is tropomyosin. Tropomyosin will be lowered. Okay, now we're getting into the whole cross-bridge thing, okay? Which protein had the heads? Myosin had heads, can now contact which protein had the active sites? Which protein had the active sites? Nope. Somebody, phone a friend? Actin, exactly. Actin had the active sites. So with calcium bound to troponin, the tropomyosin was lower. The myosin heads can now contact the actin active sites. When the heads and the active sites are bound to one another, it is called a...
I knew one before that.
What is it called? Somebody raise your hand if they know. Okay, about cross-bridge, good. Okay, now I'm going to let you raise your hand. I'll call on you. I will stop torturing you individually. But you learn a hell of a lot better if you think I'm going to ask you, right? You do. You're like, oh, damn it, here it comes. Okay, so where were we? Cross-bridge. Okay, so E says to initiate a power stroke. So here we are, guys. We did the flappy. Get your flaps involved if necessary. So to initiate a power stroke, what must fall off? Raise your hand. Go ahead. Phosphate must fall off the blank head. Which protein has the heads again? Raise your hand. Go ahead. Myosin has the heads, exactly. The phosphate must fall off the myosin head. The myosin head now pivots for the first power stroke. And blank falls off the myosin head to prepare for the next cycle. ADP, good. Okay, now we're going to cross-bridge detachment. Okay, we're going to reinforce this again. What protein has the heads again? Everyone. Myosin has the heads again. Must release the, what protein has the active sites again? Actin has the active sites. And reset for another power stroke. In order for it to release, this requires ATP. You can all spell that, right? ATP. ATP. So this requires ATP. For a sarcomere to shorten, I want a hand raised for this one. This attach, power stroke, detach cycle must happen many times. This repetitive process is called cross-bridge cycling. High five. Good job. Okay, now we're going to try to relax. Okay, in order to relax, we need to stop messing with it with the nervous system, right? Does that make sense? Okay, so let's talk about that first. In order for the muscle fiber to relax, the action potential on blank must end or acetylcholine would continue to be released. Who knows what that one is? Go ahead. Yeah, axon, neuron, presynaptic cell, something that tells you not the muscle, right? That makes sense? Okay, so acetylcholine that was already in the neuromuscular junction must be destroyed or the muscle receptors would continue to detect it. The enzyme, raise your hand, destroys acetylcholine at the neuromuscular junction. Go ahead. Acetylcholine esterase. Good job. All of those syllables, all in the right order and everything. Good job. Okay, acetylcholine esterase destroys acetylcholine at the neuromuscular junction. No acetylcholine means no depolarizing graded potential on the muscle cell. And therefore, if you don't get a depolarizing graded potential, you can't get a action potential. You can't get an action potential, yes? Okay? No action potential on the sarcolemma. No action potential on the sarcolemma means that what won't be released from the SR? Calcium won't be released from the SR. Good? Calcium present in the sarcomere must be removed for the, raise your hand, what's the barricade protein? Go ahead, Travis. Tropomyosin, for the barricade protein tropomyosin to go back up. This is accomplished by pumping calcium back into the SR and requires ATP. Exactly, good. Now, one last time. The blank thin myofilaments slip back into place and the sarcomere lengthens. What is it? Actin, thin myofilaments. This worksheet is a synopsis of all of the meaty stuff that we did last time. Okay? You can get all the nouns and all the verbs in all the right places. You will do well on this portion of your test. Cool? Okay. So,